like to invite Dr. Anita Kaiser from the University of Toronto to moderate this session. Dr. Kaiser completed a Master's of Science and doctoral degree in Rehabilitation Sciences at U of T, specializing in activity-based therapy. She's an adjunct lecturer at U of T and director for research for the Canadian Spinal Research Organization. Anita has been living with a spinal cord injury for over 25 years and has mentored over 80 individuals with SCI through SCI Ontario. She's a member of the Knowledge Translation Committee for Mend the Gap Project and a partner with us whose involvement and engagement we relish and enjoy immensely. Dr. Kaiser, over to you.
Thanks very much, Dr. Illes, for that wonderful introduction. And hi and welcome, everybody. It's nice to see so many of you here. We hope this next hour is very informative for you. So the way the hour will proceed, I'll start by introducing our three panelists, and that'll be followed by each of them providing a 5-minute update on their respective areas related to the Mend the Gap project. And then after that, we'll open it up for a panel discussion and open Q&A where we invite all of you to chime in with your questions, comments, concerns that you have. You know, this is really a session for all of you to get information about the project. So we really want to hear from you about what you want to know and how you want to get involved with this project and the information that you need for that. So I'll start first with our introductions. So first off, we have Dr. Wolfram Tetzlaff. Dr. Tetzlaff obtained his MD degree in Germany and his PhD in Calgary, followed by faculty appointments at the universities of Calgary, Ottawa, and British Columbia, where he currently holds the Eddie Ehlers Chair in Spinal Cord Injury Research. From 2013 to 2023, he served 2 terms as a director of ICORD and continues to lead a research program focusing on experimental strategies for neuroprotection and neurorepair after spinal cord injury. Next off, we have Dr. John Madden. He is a professor of electrical and computer engineering at UBC and the director of Mend the Gap. John is working to scale up fabrication of tiny rods that Mend the Gap is using to guide nerve growth. John and his team are also working on pressure injury prevention devices for wheelchairs. And then finally, we have Dr. Tanya Barretto. She's a postdoctoral fellow at Neuroethics Canada and the program lead of the ethics arm of Mend the Gap. She has a Master of Science in Biochemistry from the University of Essex and a PhD in Medical Sciences from the University of Toronto, focusing on the therapeutic potential of stem cells for traumatic brain injury. She plans to use her preclinical science background in the area of regenerative medicine to advance the ethics program of Mend the Gap. So I'll have Dr. Tetzlaff start things off.
You all appreciate that the spinal cord is a highly complex extension of the central nervous system, but what people don't know, that is actually an immense number of nerve fibers, up to 10 million is estimated in the human spinal cord. Many of them get injured in a spinal cord injury, and we see about in Canada 1,200 new cases every year, and we have about 30,000 people living with SCI in Canada, which is about 1/10 of what we see in the US. Spinal cord injury means immense losses of quality of life, but also lifespan, and lost opportunities in life and costs associated. And for the healthcare system, it means anywhere between $2 million and $5 million healthcare dollars over the lifespan of the person. Current treatments are typically acute emergency to stabilize the person, to stabilize blood pressure, to optimize perfusion in the injured spinal cord area, to stabilize the bones of the spinal column, and to prevent complications, ideally prevent infections, pneumonia, and so on. After that, a person gets then handed over to rehabilitation programs, which are widely variable, and the duration of those are similarly variable. And they work if there are some nerve fibers spared, and that's the case for most people. the research has seen 3 decades of very active research in 3 major areas. One is the area of electrical stimulation, now known as neuromodulation strategies. And we hear all those breakthroughs over the last decade with episodal cases of a person. But what we don't know is in larger studies who will benefit, who will not. And it's also dependent on how much of the fibers are actually preserved to take advantage of these type of rehab approaches. We have also seen over 3 decades, almost 4.5, on neuroprotection strategies, and still we don't have a single treatment that is widely accepted as a neuroprotective treatment that is given early after injury. And there are about 20 trials going on or have been concluded in either or. And we have also seen 3 decades of research trying to coax the nerve fibers to regrow, because when a nerve fiber is interrupted, it has to regrow from that cut end into the actual distal spinal cord. Why has it been so difficult? Well, neuroprotection works in animals, typically when the treatment is given early, but that is very difficult to translate in a human setting where you really have to come within the first hour to have it effective. But we don't really know the details of the differences of the dimensions of the human spinal cord and the pharmacokinetics and the actual reaching of the drugs, which are very much challenged by the underperfusion. Nerve fiber regeneration traditionally failed. We know that since over 100 years, and there are reasons like a very sluggish growth response of the injured nerve cell itself, a scar formation, a formation of a cystic cavity, and we have a hostile environment at the lesion site. And clinically, it's very difficult to actually address all these multiple aspects in one package. In particular, what is necessary is to build a bridge over this hostile lesion site, which is then combined with treatments that address the other complexities which I just mentioned. Therefore, in Mend the Gap, we are focusing on a minimally invasive approach to build such a bridge. John, over to you.
So Wolf has shown how complex this problem is, which explains that even though we've known for more than 50 years that the axons and the nerve fibers can regrow, it's been very difficult to achieve this. And so here's an animation. As an electrical engineer, we know that I see this as the spinal cord as being the circuit elements or the cables and some of the processing that connects our brain to the rest of our bodies. And so we see that in this animation. And what happens when that is interrupted? So typically we have these tiny nerve fibers or axons that get disrupted. And in a human, that can be at a very large scale, centimeter scale. So it can end up that quite a large fraction of the total number of nerve fibers are disrupted. Even if we have the right conditions for regrowth, there's scarring that impedes that growth. And so we need a combination treatment that can deal with multiple issues. So what we've done in Mend the Gap is get together a team of experts from around the world who are experts in biomaterials. So we need a material on which the nerve fibers will grow and pass through this injured region. We need to mitigate the scar, so soften the scar to allow the nerve fibers to grow in and through it. We need to stimulate growth. And it may also be necessary to have some robotic surgery to help with the administrating the treatment effectively. So what we have come up with through this team is an approach that involves needle injection of a material into the injured region in the spinal cord. So you'll see the needle coming through in a moment. It will inject into the injured region a matrix that is favorable for these nerve fibers to grow through. And you'll also see some tiny rods that are whose diameter is about a 20th that of a hair. And we align these in a magnetic field, and their purpose is to help guide the direction of the axon growth through this region. Because otherwise, these nerve fibers have a tendency to grow in quite random directions, and it's a long distance compared to their diameter. And the hope is that that treatment altogether will lead to some degree of, as we'd say in electrical engineering, reconnection of the circuit and return of some function. If we can achieve this, this will be really the first time that this kind of regeneration has been done effectively in a human for such significant scale of injuries. So in our project, we are about halfway through. We are at the stage where each of these separate treatments have had their initial feasibility examined, and now we're bringing them together, and in the hope that this combination of treatments will make a big difference. And this is guided by our ethics team, and we have some very important considerations in making sure that we are actually solving the right problem and one that the community will accept. And so on that, I'll turn it over to Tanya.
Thanks, John. So as John mentioned, the goal of the ethics team is to align community values and needs with the scientific enterprise of work that has just been described here. And this is all with the ultimate goal that the intervention has maximum benefit for the people who live with spinal cord injury. And so our evidence-based approach entails a suite of studies that will hopefully enable us to accomplish this. And in our first suite of studies, we surveyed the literature to identify the important priorities for improving quality of life after injury. And among the many important ethics themes that were identified, the one that was really significant was empowered choice was a key value there. And this really means being able to live and make decisions based on an individual's personal values, relationships, or cultures, or autonomy, which is a very important ethics concept. So what we did was we looked at how this concept is discussed in the literature. And what we found was that it's discussed in a very inconsistent manner. So for example, some papers make references to functional independence and capacity for self-care, whereas others discuss this in the context of cognitive capacity. And what's really important here is contextualizing the concept of autonomy in SCI research, as this will really limit bias and empower people to make choices that align with their values. So now a key factor to empowering choice and informed decision-making is having access to information. So what we did in our second suite of studies is we conducted a study to look at how SCI research is communicated by the traditional news media. And what we found was that news media reports tend to communicate research findings in a balanced way. And that really means that it highlights both the risks and benefits of a study. But the media also tends to focus on or discuss how these advancements in research are in relation to how many people would have access to them or benefit from these treatments. Another major finding from this study was that there's a drop-off in media reports on spinal cord injury research after 2016. And this is a gap that really needs to be addressed. And this responsibility is shared by all of us, so researchers, institutions, funders, and the media. And this is to ensure that the lines of communication remain open and that the information we are putting out remains up to date, and so that the research goals align with the values of the community. So then what we did was in our second study in this suite, we looked at how the public responds to these research studies. And the data showed that the public are actually very interested in the research information and the progress. And this really highlights that, need for sustained communication. But the public also wants to know how many people would benefit or be able to access these treatments. And these are some things that we keep in mind at Mend the Gap as well. In our third suite of studies, we looked at the inclusion of people living with SCI in different avenues of research. So in the first study, we looked at the representation of people living with spinal cord injury in the peer review and editorial process, so that they have the ability to comment on research that pertains to them. And the data show that there's generally a positive trend towards inclusivity, but more work needs to be done. The primary challenge that was highlighted is identifying people living with SCI or disability so that they can participate. We also looked at the inclusivity of people living with SCI in clinical research and found that women with reproductive potential are overwhelmingly excluded from participating in trials, and those living with mental illness or substance use are also excluded. And these exclusion criteria, they are not completely in line with the ethical guidelines for research and really need to be addressed. So as you can see, we are hard at work as a community, but we still have a lot of work to do to improve communication and representation to continue to align research goals with the community values. And the ethics team works really closely with the Mend the Gap scientists, and we also work with the Knowledge Translation Advisory Council and our Ethics Advisory Board to guide our studies so that we are on the right track here. And finally, I'd like to share that we are, currently laying the groundwork so that when we do engage with people living with SCI, we do so in a meaningful and inclusive manner. With that, I will hand it back to you, Anita.
Thank you all for providing that update on each of your areas. So I'll start with kicking things off with the open discussion and Q&A. I'll just remind our attendees in the audience that if you have any questions that you'd like to ask any of the panelists, feel free to either raise your hand or you can add it to the chat, and I'll toggle between them and try and get to as many of them as possible. I am going to reserve a few minutes at the end for a poll that we'd like to send out to all of you to get your feedback on some of what you've heard today. So I'll start off with my first, I think, basic question is just for all of us to know, like, who is really the target population for this intervention that you're proposing? Complete or incomplete injuries, acute versus chronic, and is it open to individuals who have traumatic or non-traumatic injuries? So Dr. Tetzlaff, Dr. Madden, I think this is more directed to both of you.
Yeah, it's a very complex question because you are addressing really a very important one. It is guided by what is feasible in the animals first. And that would mean we would essentially envision in the animals a treatment interval between 4 and 6 weeks. And that would mean clinically we would probably target it initially to motor complete. That's about 40–50% at around 1 to 2 months post-injury. And these are traumatic injuries. But obviously, once this works, one would then like to extend the time frame into the more longer intervals, but also towards the shorter intervals to combine it with neuroprotective treatments. But for the time being, the feasibility of an injectable hydrogel in our animal studies suggests the optimal time is here several weeks after the lesion. That's when you have a formation of a cavitation and so on. That's a detail which you don't need to know yet.
So thank you for that. I'm just thinking about the fact that, majority of the community and people who are living with a spinal cord injury are in the chronic stages of their injury. So how could a project like this, benefit them?
Very, very, very true what you say. And what I'm saying is the attempt should then be made to see whether chronic lesions can be modified in a way that allows such an approach to be feasible even after 6 or 12 months or longer after injury. That is completely unclear at the present time, but it should never be left out of the focus and out of the scope. But we have to start somewhere, and we think we should start where the feasibility seems to be best.
Okay, thank you. And I've just been told that the chat is disabled, but you should be able to use the Q&A. I don't have access to that, but I think Dr. Judy Illes will be helping to support that. So just let me know if there's any questions that come in through there. But you should also be able to raise your hand if you have any questions, and we can have you unmuted as well. So my next question would be directed to you, Dr. Barretto. What's next for the ethics team?
Thank you for that, Anita. So, one of the big things that we're working on is disseminating a national survey to those living with spinal cord injury to ask them about their, opinions and their perspectives on the Mend the Gap intervention that we're discussing here. So, I would encourage the audience, like if you've got any key points that you think we should be asking, please put them in the chat or in the sort of the Q&A, and we will also be sending a feedback email where you can, reach out to us. So that's really what we're working on, is to start to get the opinions of the SCI community at large.
Mm-hmm. And one of the things that you talked about in your update was about the area that you've been focused on, which is empowering choice. And I'm assuming that will probably get addressed in the survey that you plan on launching. But what are the ways that you're looking to try and address that and tackle that issue?
Yeah, so we is to provide the right information at first. That's really what we are hoping to do, and then there will be questions very specific about the intervention, so that we will hope to incorporate into the science as much as we can, and to the best of our ability. And there will also be sort of, open-ended questions where people can give their perspectives on that. And so really to try and make sure that we are aligning this intervention with the values of people living with SCI.
That's great.
I see a question, Anita, from Dr. Main and Mr. Meklovich. I don't know if you can see their hands up as well, so.
No, I didn't. So thanks for letting me know. Go ahead, Maskovich.
You're still on mute.
Yeah, I was just trying to unmute. I have a couple of questions. There's a couple of seminal papers, one by Samuel Stoop, I think in 2021, from Northwestern. And then there was a more recent paper, which I think was 2023 by Square out of the Lusan group. I'm curious on your perspective on that. As well, I believe NerveGen was undergoing this summer, I think was in the middle of a phase 2 trial in Chicago. They'd asked for volunteers. So in consideration of those three items, could you make some comments? Particularly Stoop's paper.
Yeah. This paper looks on the surface very good and very interesting. And we tried to get in contact with Stoop to see whether we can collaborate and compare it to our material side by side. Stoop is not open to outside Chicago collaborations. We have to see what is actually now reproduced in the hands of others. And we have to wait how this is surviving the actual replication phase. It's a very interesting approach to deliver in a hydrogel fibroblast growth factor fragments that are activating this particular component. We are also in Mend the Gap discussing exactly a similar approach with our hydrogels, where we're actually delivering such peptide ligands to the fibroblast growth factor here, and we will see what happens. The Lausanne paper was it was really mainly about stimulation and the understanding of the spinal cord injury, and it's very interesting what this group is doing, high quality work, and they are really the leading team on the neuromodulation approaches. And in some patients, these neuromodulation approaches are quite effective, but the word is really out who will benefit and who will actually respond to it and how quickly. And then it becomes a huge a question of resources, what jurisdiction can actually afford to do this, and what are the criteria for the patient enrollment for those things. NervGen is an interesting component. We collaborate with NervGen. They are part of our package, so to say, or we are part of theirs. We try to keep in close contact. We are seeing some sprouting growth. Dr. Karim Fouad did that in the Mentegam team for the NervGen component, and we hope to integrate it into our ultimate treatment. And we are quite pleased to see NervGen going ahead with human subjects to look at safety and toxicology of that compound. The actual trial in humans would not allow any conclusions. It's too early.
Just to put it in perspective, for Stup's treatment, we could imagine that that would be part of the injection that you saw in the animation that could be injected into the lesion, either on its own or with some of our rods, or and with other treatments.
How does one get to be a participant in your studies? I'm 75 and I keep getting bumped out. I notice every time they have an I-cord study, they chop it off at 65, the exception being the one on the external stimulation that Dr. Krajikov, I participated in that. So although I'm 75, I'm in good health. I had my accident 10 years ago, and I'm very interested in being a participant. So why do you cut us off at 65? Seems to be a human rights issue there as well.
I don't want to answer this question in your particular case, but when you go to clinicaltrial.gov, this is the common practice for clinical studies, typically at 65, 70. I completely share your feeling because I'm 70 myself, and I completely agree with you. One should allow and consider that with the appropriate control enrollment, and I share your frustration. But this being said, you have to understand our treatment is not ready in any regards for chronic treatment, and therefore, let alone we are not ready for the acute either. Therefore, we are not able to actually engage with you closer and say, we want to trial you. So that wouldn't happen yet. And this may be years ahead.
As you can see from our presentation, this is a complex process with lots of parts. And so what we are hoping is that by the end of our 6-year project, we will have enough data to justify going ahead towards trials in humans.
Thank you both. Dr. Main, you had a question?
You're on mute, Alicia.
Great.
Nope, sir, you're still on mute.
Is it okay? It didn't give me permission to unmute, so that's what my my bad pictogram was. Thank you. Great discussion, and I'm also excited about the survey and the opportunity with that. So my question is, what breadth of population, to the question about acute versus chronic, patients that are living with spinal cord injury, to what group of that are you intending to send the survey? And the second is, have you considered, beyond this specific intervention, which is, right now, mainly for acute patients, have you considered trying to take the opportunity to ask about preferences for some other types of interventions? And one that I have in mind that's very different, but also relevant to people in chronic case of spinal cord injury, is the new Health Canada approval of the human in motion exoskeleton, right? Which is quite exciting. And, it now has FDA approval. It's now going to be available in, in treatment facilities, hopefully in Canada. So is there an opportunity, my question is to you, to, depending on which group that you're sending the survey to add a couple of questions that are a broader set of things that could bring hope, and ask them about them?
Yeah, I think, sorry, I was just going to say there is that option for sure. But I think, really we want to also be able to tailor the biomaterial intervention really to to this cohort, right? So if we make it too broad, I think that just goes all over the place. But Dr. Tetzlaff, if you want to.
Well, I think the discussion whether there has to be a specific rehab regimen that may or may not include an exoskeleton is a valid one. But as you just said, Tanya, the focus is initially on the injectables with some co-treatments addressing the other complexity of the problem. And it's very clear that no treatment that tries to promote growth can neglect the addition of rehabilitation training. And the exoskeleton could provide one of those means to that. And it is for us at the moment too early, but the nice thing is there may be other things coming along in a few more years, which we may then want to put into this rehabilitation envelope. And for sure, electrical stimulation, neuromodulation is another one. So thank you so much for that question. You're absolutely right. It has to be considered, but we're not quite at that stage yet.
Thank you. So I'm going to answer some of the questions that are coming in from the chat. There's one that's coming in from Paul that says, I missed the beginning of this webinar, but wonder if the current interventions like steroidal injections during the acute phase to limit scarring is still used and to be combined with hydrogel injection.
I can quickly answer it. It's optional, depends what the doctor who is treating you considers appropriate in your particular case. You don't do it in complex injured people that have other open wounds and other situations in which metoprolol can be actually damaging and has risks of pneumonia and so on. But it's not standard of care. If the physician decides that you are not the case for that, for this and this and this reasons, or you don't fit the time window and so on, you may not get it. The combination with the Mend the Gap, there may be better neuroprotective treatments by that time. And yes, as I indicated, I like to push in our preclinical work the timeline also to the more acute side as well, once we have figured out how to deliver it in the more acute time. And that is then appropriate to incorporate it with the gels or and with the biomaterial that we are using. So the short answer is a neuroprotective treatment such as metoprolol is probably at some point appropriate if these acute deliveries work.
Thank you. So there's another question in the chat from Vivian Garcia, and she asks, if there is a problem in a body after the gel is inserted, can it be removed? Will that be a part of the research?
The short answer is the gel will not be able to be removed. It's slowly degrading. It is, for all we know, in other parts of the body, highly biocompatible. And some of the research actually suggests that, for instance, polyethylene glycol in some forms is actually protective. Some people use it to fuse nerves. So the short answer is no, but we don't expect major side effects from that. This being said, we need to confirm that in our own hands in our long-term experiments to address safety toxicology. But for that reason, we are very reluctant to use materials that have a suspect risk.
We're trying to be very conservative in our selection so that we're starting with materials that have been largely approved for use, at least in other parts of the body.
Right.
And Dr. Illes, I can see you have your hand up.
You're muted.
You're muted.
Why don't we take the question from the anonymous participant, and then I'm happy to jump in with my question.
Okay, so another question from the chat says, how do you prevent further damage from being inflicted on the spinal cord during the injection process?
Brilliant question. We have to exactly work on that project because those interventions have exactly that risk that you are producing an additional pressure or anything like that. We have seen it and also in other trials of the in vivo therapeutics trial, for instance, where scaffolds were inserted. And we have to find out exactly what volume is in that lesion necessary, image the lesion, appropriately address the right amount here, and make sure that we are targeting the actual cavitations in the spinal cord in order to avoid damage to preserved structures that are lying next to it.
So this is one of the reasons that we're using needle injection to minimize the potential for damage, but there always is some potential, and that we're considering using image-guided robotic surgery to help inject in exactly the right location within exactly the right volume.
Go ahead, Judy.
Thank you. And then I see we have a few more questions that have come in on the Q&A. My question is has to do for others actually who are participating in this event. And this is one of the reasons we call it a conversation, is that we can benefit from the expertise of those who have joined us. And I don't know if there's anybody with us from Health Canada, but I do see Zach McKinney from FDA. It's a big day for you in the United States today. And I'm sorry to put you on the spot, but I would like to know and hear, how do we best approach the regulatory space for the kind of biomaterials that Mendegap is trying to develop to ensure that we maximize efficiency and really getting a successful product to the patients who it's intended to benefit. So if Health Canada, you're with us, please jump in. Zach, if you don't mind jumping in, we would really appreciate hearing your input.
Thank you for the invitation, Judy, both to this event and to comment. Yeah, big day indeed. We're processing a lot down here on a lot of fronts. I'll leave it at that. But to your question, I think it's a very good one, actually, because as a theme that often arises, among this patient population is regarding, the necessity of, multi-pronged or even multimodal treatments that include, aspects of, either, pharmacological interventions plus, biomaterials plus, some combination of rehabilitation, whether it's robotically mediated or neuromodulation mediated, and figuring out how all of those risks and benefits interact certainly is, I think, an open area for exploration with regard to injected biomaterials in general. So, I mean, I am a lead reviewer with OHT5, which is our Office of Neurological and Physical Medicine Devices. We cover spinal neuromodulation and exoskeletons. But those materials would certainly get reviewed by a different office within FDA, and so discussing, I think that question specific to those materials would be better directed to that office, and I don't think I can offer any terribly useful comment at this moment. What I can say though is that given the number of variables in play, that clinical trial design, in order to correctly identify, which are the the important variables, I think will be of interest to us. And part of that also is going to be, what are the outcome measures that are the most meaningful, because it's clear that there are a lot of potential benefits, of the use of exoskeletons, and not all of those benefits are clearly represented in the indications for use statements of clear devices. So essentially, I think the short answer really is come talk to us in a Q submission. They're free, and this is a dialogue we would love to have. And can't say anything definitive now, but we would love to work collaboratively with the research community to navigate these waters.
Thank you, Zach. So there's another question that came in from the Q&A from Jen French. She actually has two questions, so I'll ask the first one and let you respond, and then ask the second. So the first one is for Dr. Madden. She said, mentioned feasibility of this intervention. Has this been safety tested with humans? And if not, what animal models have been used for translation?
It has not been safety tested with humans, not the whole the complete package. A number of the materials and drugs that we're using have been used in humans in various forms. And so we're building on that, but our initial tests are all in rodents, and our next phase is to look at porcine models.
Thank you. And then our second question was regarding the community survey. How will you include people with SCI lived experience in the design of the survey rather than only the distribution of the survey?
I will take that one. That's an excellent question. So currently we are just working with SCIBC and Praxis in the development of that. But part of this conversations is to put out there that if you are someone living with spinal cord injury and would like to, have your say in how this is developed, please reach out to us and we will try and set up some kind of event or focus group where we can discuss this. And that's what we want. We really want to include people living with SCI in the development of the survey as well. So.
Yeah, we'd love to get your feedback in the survey as we complete this session as well.
Yes.
And I think Wayne has his hand up.
Last question for me. There's a paper that just came out last month from Wang et al. Looking at CART therapy for neurodegenerative diseases. They were able to reverse MS in 3 patients. Do you foresee this, or can you see this as potential application for SCIs?
I have not seen the paper. I have to apologize. I'm not following every paper in the MS field. I will happily go back, and I cannot answer the question. Sorry. If you can be more specific, I can give you a hint, but I don't know.
If you want, use my email to me, and I'll send you the paper.
Sure.
Thank you. So there's another question that came in the Q&A from Paul. With hydrogel injection, the site of the injection and resulting neuroregeneration is not something to be observed since it is microscopic and requiring direction to actually see. So I wonder if it would be interesting to try this injectable hydrogel with an observable CNS tissue in the eye, the retina. Disorders such as macular degeneration could have hydrogel injected, then healing observed.
So I'll let Wolf answer the question, but we do, one of our members of our team, Alison Fournier, does have an optic nerve crush model that we are considering exploring. I'll just mention that we are not able to directly observe axon regeneration as it happens, but we can look at it in animals after the experiment by doing sectioning. So at least we are able to see it that way. But I don't know, Wolf, if you have more thoughts.
It's very difficult to see axon regeneration in humans unless you have large bundles that are inferred to be axons on MRI by getting a longitudinal vector signal. But proving that these are axons is essentially not possible, despite the fact that sometimes people claim these are axons. The axons are much smaller than the resolution, so they're usually a few microns thick only, and the resolution of MRIs are tens of microns. So we are actually talking another magnitude of order of magnitude here. So that's the short answer. Doing it in the eye, Molly Scheukert also is using one of the gels in the eye for that reason, but not seeing individual axons. This is not feasible, and the short answer is we are not doing much beyond the work that just John mentioned in the optic nerve with Dr. Fournier.
Thank you. I'm not seeing any more hands up, so I will go to another question in the Q&A. This is a great one. If this process doesn't work, what is the next step for individuals with SCI?
So first of all, if our project, even though it's very high risk, and there's a good chance that our complete project won't all come together. We already have some promising leads on scar treatment, on growth treatment. And so, although those also have to go through a number of additional steps on their own, there's a chance that those aspects and also further developments in robotic surgery and so on will have an impact and will become part of the standard treatment in SCI. What happens if this doesn't work? If our overall treatment doesn't work, I think there are different routes that are being explored. One is a more electronic approach where you imagine that above the site of injury, you're making recordings of what's happening in the nervous system, and below the site of injury, and that information is then bypassed electronically from above and below. And so that technology is still quite crude, but still shows some promise. So that's one avenue. There's still interest in implantable scaffolds that help support growth. Wolf can comment on this further, but it seems like the results so far are not strongly supporting this. This is basically implanting what Wolf calls a cigarette filter shaped device that allows for the nerve fibers to grow through channels. And so it's more invasive than what we're proposing, but also is further along in its demonstration. And then maybe, I don't know, Wolf, if there's anything additionally you want to add, but I think as the field advances and we're learning more, I hope that there will be other combination efforts like ours. But to do that requires a lot of resources and vision from government funding to get it launched, so we can get it to a stage where it's then ready for implementation in the clinic. And we were fortunate enough in this case that the NFRF Transformations Program was willing to fund researchers not just in Canada, but also bring in world experts so that we could approach this with the best possible team and achieve the best possible success, understanding also that there's a high risk of failure.
Now, I'd like to add that has been in the spinal cord field quite commonly, so that trials failed because of the complexity of the problem. This being said, that should not hold us back to further refine these hydrogels in their modifications, we call them functionalizations, to make them better with regards to the way the nerve cells see them, with regards to their ability to leach out and secrete drugs, and thereby slowly improve it. In our tissue culture models, we would go ahead first, modify them accordingly, and then essentially try this again in our in vivo models. So the short answer is research never stops. You never take failure as an end of a research line. You take failure as something to learn from and interpret where your what failure was, and then modify and go ahead. And so the short answer is we have a lot of work to do, and we don't know at what point we have enough data to say, yeah, that's the time to translate now, because even from there will be most likely more and more improvement. This is the nature of the situation, in particular when you have such a complex organ like the spinal cord.
Great point, Dr. Tetzlaff. So we have one last question in the Q&A. What types of animal models have you been working with so far where efficacy for this treatment has begun to be proven? Following up on that, what are the biggest challenges you foresee with scaling up to humans, considering the anatomical differences between species and patterns of nervous system recovery?
Our data are mainly in rats, and we are working mainly in rats for multiple reasons. It's an animal where we can do testing as well as anatomical analysis, and as well as the delivery into a lesion site. But of course, as John was mentioning earlier, Dr. Madden mentioning earlier, that pigs are the intermediary on our translational efforts to humans, because the dimensions of the rat spinal cord being only 3 millimeters, a human spinal cord about 12 millimeters, are hugely different and more challenges. So the short answer is we are having to go this step of wide escalation, in particular with such a complex treatment.
I just want to extend a thank you to all of our panelists and for all the wonderful questions that were coming in. It was a great discussion.
It is the top of the hour. I want to thank everybody so much for this extremely interesting and interactive community conversation. It's exactly what we were hoping to achieve with you. We ought to do it again soon. Anita, Dr. Kaiser, thank you for moderating for us, and to the KT team for supporting this event. Dr. Tetzlaff and Dr. Madden and Dr. Barretto, thank you so much for being our panelists. And most importantly, thank you to the 90 of you who joined us today for being part of this conversation. Have a very good rest of the day and take care.